So I think we can start now. Welcome to all of you to our Tech Talks at Rocket. With this webinar series, we would like to give you some information about the creators behind our innovation projects within the Rocket Reloaded project. And today we focus on a very interesting project, which is called SensiChip. It's about DNA sensing chip for detection of infectious disease DNA markers in aquatic systems. But first of all, I would like to hand over to my colleague Alex van Geldrop so that he can give a short overview about Rocket Reloaded. So Alex, the screen is yours.
Thank you, Hendrik. So can I just take over the screen? So a really short introduction from my side so that we can quickly move on towards the actual content. Like Hendrik mentioned, the SensiChip project, which is one of the projects that is going to be explained in a few minutes, is part of the Rocket Reloaded project. And Rocket Reloaded, you should be able to see my screen right now.
Yes.
Yeah. Great. Rocket Reloaded is an Interreg 5A program, which means that it's funded by the European Union, and that the main goal behind all of the projects within Interreg is to stimulate Dutch-German collaboration. And the way they are doing that in this particular case is by stimulating innovation projects between Dutch and German SMEs that create stronger ties between the two companies, between the several companies around both sides of the border. And it is also good for the competitiveness of the region as a whole. Within that Interreg project, we developed two programs, Rocket and Rocket Reloaded. Rocket has already run from 2016 until 2020. And Rocket Reloaded is the successor, which is currently running and will end in the middle of next year. And both of these programs had several innovation projects within them. And all of these innovation projects were focused on key enabling technologies, such as nanotechnology, photonics, microelectronics, innovative materials, etc. And there was all, in each of these projects, there was a link between Dutch and German companies. So that's the goal that we have with these kinds of projects. One of the 6 projects within the Rocket Reloaded project is SensiChip, where we'll get in a minute. For those of you who are interested in some of the other programs, the other projects that are running, there's a website, www.rocket-innovations.eu, where you can find information on all of the projects that are running and that have run. For the projects within Rocket, there have been animated movies that give you some insight in what had happened there and what kind of companies were involved, what knowledge institutions were involved, so that you can see which companies you might address if you want to innovate something yourself, or if you want to collaborate across the border. And these programs do not run themselves. They are developed, supervised, coordinated by a number of partners. Those are also some of the partners that are present today. As also know, we are the lead partner of the project, but we do this in close collaboration, in this case with NMWP, the nano-micro-werkstoffen Photonica cluster from North Rhine-Westphalia, Holland Semiconductors, which is a part of HiTechNL, and the TAF Münster, and our associate partners who were full partners in Rocket, and because of some geographical locations, now associate partners in Rocket Reloaded, Innosperlig and Novelty, en de Brabantse Ontwikkelingsmaatschappij, de BOL. So our call to action: if you are looking to bring an idea into an innovation, and you're looking for cross-border collaboration or in general for support, then these partners are willing and able to help you bring it to the next step. So if there are ideas and you're looking for support, feel free to contact any one of us, the most relevant partner for you, and we'll try to assist you in bringing it one step further. That's it from my side, just to give you a bit of an overview of the framework in which the project was developed. Now onto the content, and I think Floris van den Brink will be the first one to take the word, or at least somebody from the SensiChip project. So I'll give the microphone to the next person.
Yes, so I would say, Floris, I think you will start possibly. So now the floor is yours to the SensiChip team.
Yes, okay. Welcome everybody to the presentation of our SensiChip project. This project was carried out over the last 2 years with 4 partners. From the German side, we had the Tunatech. Tunatech will actually start this webinar, so I wasn't prepared at all to get the floor now, but in any case...
Sorry for that.
No, that doesn't matter. And the other 3 partners are Nitor, Lyonix, and Curin. And in this order, they will be presenting this afternoon, all about 7 to 8 minutes, after which all the floor is open for questions. So, well, Florian, then I'll give the microphone to you.
Okay. Okay, I try to share my screen. Did it before, I hope it will work again.
Yes, perfect.
Okay. Okay, welcome everybody. My name is Florian Brutter. I'm one of the partners from Tunatech, Germany. And I would like to give you a short introduction about Tunatech's work and in general about the SensiChip project in the Rocket Reloaded framework. As Henrik said before, the SensiChip stands for a DNA sensing chip for the detection of infectious disease DNA markers in aquaculture. So Tunatech in this collaboration is let's say the biological part, and later on via Nitor to Curin, it becomes more technical. So what was the aim of the project? I mean, why aquaculture and why DNA chips? And okay, I try to give a very short explanation, because early pathogen detection, or maybe even real-time detection later on in recirculating aquaculture system is quite nice with the help of eDNA to have a technique to improve sustainability and productivity. So it would be quite nice for future aquaculture, which will be mainly on land in recirculating systems, not only juvenile production, also grow out for adult fish, to easily detect and early detect pathogens in these tanks, to prevent breakout of diseases, or to use antibiotics wisely and not just put a bunch of antibiotics in your system. So that is the aim of the project. Okay, and what Tunatech's aim was mainly in the work package 2 and in the work package 3. Work package 2 was the biomarker identification for the diagnostic applications. And therefore Tunatech performed kind of a small market analysis. We had talks with colleagues from the aquaculture, especially from the salmon sector, which are the diseases or the pathogens of their interest. And after the talks, Tunatech had chosen 6 different bacteria and 2 viruses, and we have them chosen due to of course the responsibility for economic losses in salmon aquaculture. As I said, we mainly worked in salmon aquaculture because salmon aquaculture is the biggest aquaculture sector within Europe. We have chosen them due to their safety and risk group, because at Tunatech we are only able to cultivate organisms or bacteria of the risk group 1. Then, of course, regarding the availability, of course, we have to obtain, we have to purchase these strains. And then we selected, well, we would like to have a selection of gram-positive and gram-negative bacteria, because sometimes some are easier to handle, and therefore we'd like to have both of these bacteria strains. Right down here at the bottom, you can see the selected 6 bacteria strains and the 2 virus strains. I won't go through them all, but all of these bacteria strains, they are the causative agent of a specific fish disease. For example, the Aeromonas salmonicida bacteria is a causative agent of the furunculosis disease, which is a major disease in salmon or aquaculture of salmonid fish, which means salmon, trout, char, etc. And it caused a lot of losses. And of course, aquaculture companies had to spend a lot of money for not only for diagnostic, but also to prevent breakout of these diseases. Regarding these two viruses down here at the bottom, we at Tunatech weren't able to get these viruses due to biohazard restrictions, but our partner NITOR was able to purchase synthetic G-blocks, which later on acted as synthetic DNA for the setup of the qPCR and also for the chip validation. Okay, what did Tunatech do during the project? As I said, we purchased or we organized different strains of bacteria, and we cultivated them in our lab. Therefore, we had to adapt some of the cultivation conditions because most of the bacteria, of course, they're living in cold water at salmon, so at 10 to 15 degrees. We increased temperature to about 28 degrees. So we had less waiting time for the bacteria to grow. After the growth of the bacteria, we determined the cell number by spectrophotometer and by simple cell counting, which was important due to the cell number which we put into the DNA extraction later on, and that we have a quantification later on the qPCR or on the real aquaculture samples, how much bacteria are in the samples. Next to just the cultivation of these bacteria strains, we also performed a PCR of the 16S ribosomal RNA gene with universal primer set with subsequence sequencing and sequence alignment with NCBI database. Just as simple because we would like to check if our strains, if our bacteria were pure bacteria strains of the bacteria would like to have, and just to hand over to our partner NITOR and Curin pure DNA extract. I think we found one contamination during the time, but most of the time it went quite well. Okay, in work package 3 and 4, the sample processing and PCR verification and proof of concept of the Photonic biosensor. Tunatech supplied lots of DNA extracts. You can see down here in total, Tunatech supplied 159 DNA extracts to usually directly to NITOR, but also to Curin. And I've grouped these DNA extracts into these 4 different groups. Left here on the top, first one, of course, are the pure bacterial DNA extracts of the named bacteria. These pure DNA extracts are, of course, for the validation of the setup qPCR by NITOR and later on also for the validation of the biosensor. So does my qPCR, or does the sensor really measure these bacteria which should have been inside the extract? The second group was have been a different dilution series of DNA extracts of the above named bacteria. So we cultivated the bacteria, counted the cell number, and extracted certain amounts, did a serial dilution, for example, from 4 million to 4 cells in buffer or any other liquid, which later on acts for the sensitivity test of the qPCR and the biosensor. The next group was were DNA extracts of fish, mainly muscle tissue from different fish, from salmon, amberjack, yellowfin, sturgeon, which acts as matrix DNA and also as control DNA, because of course we would like to specifically measure only the bacteria and of course not the salmon. And we also extracted DNA from a healthy salmon that we had a background, so a matrix DNA, from healthy salmon, which then we spiked with also a certain amount of pathogens, also a dilution series, also approximately the same numbers, 4 million to 4 cells, spiked with salmon muscle. Then we did a DNA extraction, and then we had a mixture of lots of salmon DNA and just a small part of pathogen DNA, and then NITOR tried to detect the pathogen DNA inside these extracts. Then we were also able to get some real aquaculture samples from 2 different non-commercial salmon hatcheries here in Germany, in Northern Westphalia. These were extracts from diseased salmon, which were naturally infected with A-cell, F-col, and Virus. So these were dead fish sampled from state agencies, which we were able to get. So in this case, we were able to provide real aquaculture sample as well as, let's say, spiked lab samples for the measurement at NITOR and curing. And the last group of the DNA extracts was an eDNA extract. As I said before, eDNA is environmental DNA, which means free DNA, usually meant free DNA swimming in the water from any organism, also from humans if you spit inside the water or something like that. Of course, it's a very good technique because it's not invasive. We do not have to harm the fish. Therefore, we go on with the eDNA. Therefore, we collected lots of water samples from hatchery nurseries, from salmon hatchery nurseries, of course, stream water from inflow, outflow of aquaculture facilities, and filtrated in each batch 250 milliliters of water through these filters. And then you can see, of course, it's quite dirty, and we did a DNA extraction with different methods to extract DNA from all organisms captured on this filter, and handled these extracts over to NITOR. And as well, we did some dilution series. We spiked water samples with a certain amount of living bacteria, filtrated them through the filter, and later on did the DNA extract. Okay, what TUNATEC also did, as we integrated these different eDNA extraction methods in TUNATEC, we set up different isothermal amplification protocols, which means LAMP assays. As we were working with eDNA, we widened our project scope to this LAMP assay, and LAMP is an isothermal amplification of a specific part of a specific target DNA of a relevant organism you would like to detect in water. And therefore, we designed specific primers, for example, here in this case for salmon. And if the target DNA is present in the sample, of course, only at a certain amount, we get a pH change and the color change from pink to yellow. And within the project phase, TUNATEC set up, as I said, these LAMP assay for Atlantic salmon, then for the calico crayfish, an invasive crayfish species here in Düsseldorf, and the capital of Düsseldorf would like to detect where are the crayfish. We also did it during the project, and for Aeromonas salmonicida, but this test has to be verified because there are lots of bacteria which might give a false positive result, and we have to verify it yet. And at the end of the project, TUNATEC had offered the different setup multiplex qPCR by NITOR to different state agencies and national agencies which are responsible for the detection of notifiable fish diseases in aquaculture, and also to their contracted laboratories. Okay, that's from my side. Thanks for your attention, and I think I can handle over directly to Lydia, or do we do a question stop here?
As you like. Possibly I would suggest to hand over to Lydia, and then we can have, if we have the complete view, we can ask the questions at the end.
All right, I will start share my screen. Oh, can I share my screen?
Yes, it works.
All right, perfect.
Perfect.
Let's go back to the beginning. So my name is Lydia van Laar and I work for NITER, and I was responsible for the development of the qPCR assays and also the probes for the chips for curing. I think a bit similar to Tunatech, we were also involved in work package 2 and 3. So the design of the probes and the qPCR assay, and also the field validation of the samples that were offered by Florian by Tunatech. So based on the different pathogens and viruses that were selected by Tunatech, we've designed the multiplex qPCR assays for all these pathogens and viruses. We were free in how we organized those multiplexes. So in the end, we chose for 3 different multiplex assays. So we have one multiplex qPCR assay detecting Aeromonas salmonicida, Phagococcus salmoninarum, Flavobacterium, and Columnaris, combined with an internal control. The second multiplex qPCR assay is able to detect Yersinia ruceri, Renibacterium salmoninarum, also an internal control. And the third one is a virus-based qPCR assay detecting the infectious salmonella virus and also the infectious pancreatic necrosis virus. So our internal way to go is, of course, first start with a literature study, then design the primers and probes, test them all in our lab based on synthetic controls, and when all of that looks good, we continue verification and prototyping. One small remark regarding the development of this assay and the design is that for ISAV, we were able to only detect the presence of the virus in general and not the specific pathogenic variant, but that can be done in the future potentially. But this one was quite a hard one to develop, and it might even not be possible. So we decided together to focus on the general detection only for this virus. So as Florian already mentioned, it's really important to be able to detect these viruses and pathogens at an early stage. So our goal was to develop a qPCR assay that was as sensitive as possible. So we've tested the sensitivity of our assay on synthetic controls. We did that based on the dilution series in which we detect 100,000, 10,000, 1,000, 110 copies per target. As you can see in this table, with our qPCR assay, we get a CQ value for each dilution series. It's also visible here in the amplification plots. So when you dilute it 10 times further, you see that the detection is getting a bit late, but even at here, the 10 copies is detected. So this all looked very sensitive for all the different pathogens and viruses. So we expanded this dilution series with a lower limit of detection assay experiment in which we've tested not only the dilution series again, but also multiple copies of 50, 20, 10, and 5 copies per reaction. I've summarized the sensitivity for all the different targets. So it was between the 10 copies and the 50 copies per reaction. So this is all based on our synthetic controls. All these multiplexes seem very sensitive. And with this information, we've also designed the probes for curing. And then, as Florian already explained, we received a lot of samples for validation tests. I'm not going into detail what those samples are. I think Florian already explained it quite well. So I'll continue with the results. So the most important samples for testing were the cell culture extracts, in which we saw a good detection for all of the pathogens that are present in our first and second multiplex. So they were all detected with really early CQ values. So again, sensitive detection. For one bacteria that was cultured, we had a a false positive signal in Rook. And there were 3 different pathogens detected as a negative control, and they didn't show up in any of these multiplexes. So there was no cross detection for these pathogens. The second sample set, also really important for the validation, were the extracts of the sick fish. So it's good to keep in mind that these extracts or these fish were determined sick by an external lab. However, the fish that we got are not the exact same. So we didn't receive any extracts, but just another fish. And we've tested them with our multiplex assay. And the ones that were diagnosed with ASAL and Rook were also confirmed positive with our qPCR assay. So that was a good correlation with their results. However, the fish that was diagnosed with Flavobacterium columnaris was negative in our assay. And we're not sure whether that was due to the different fish that we got or whether it was an assay error or a design error even. We've also tested the different extracts of salmon tissues, so all the heart, liver, spleen, mucus, and feces samples. We were able to detect some weak positive results in different samples, mainly in the feces, a few filter and also mucus extracts. Of these samples, it is uncertain if these positive signals are true positives or false positives. In our entire assay development, we had never seen any late signals or non-specific signals, so they seem true positives, but we were not able to confirm those results by an external lab. The same was seen for the extracts of the water and filter samples. The samples that were spiked by Florian with ASAL and VSAL were detected with our qPCR assay quite strongly, since a lot of cells were spiked, I think, in that round. And for some samples, there were again late positive signals seen, but we are not sure if it were true positives, false positives, or contamination. And then we had different dilution series in tissue or in buffer. We saw for Phagococcus salmoninarum that the sensitivity was higher compared to Aeromona salmonicida. So for Phagococcus salmoninarum, we were able to detect only the addition of 4 cells to either the tissue or the buffer. And for Aeromona salmonicida, this was 400 cells. We didn't see any differences between the tissue samples or the buffer samples, so the sensitivity between those two seemed similar. And for Rook, this same experiment was performed later, and we also saw a sensitivity of 4 cells that we were able to detect with our assay. And then we had some original water samples that were filtered and later spiked with different concentrations. The sensitivity that we saw from these spiked samples was similar to the dilution series that I just presented, but no signals were detected in the flow through of these water samples. So that means that either it was too diluted, so the assay was not sensitive enough to detect those 1 or 2 cells, or they were all stuck on the filter. And then the overall conclusion. So for our work packages, we were able to design the primers and probes for the biosensor and the qPCR assays. The qPCR assays were developed for the selected biomarkers, and yeah, we did quite a lot for the field validation of these markers. However, I think for a diagnostic setting, an extended validation set is needed for the pathogens and mainly for the viruses, since we didn't get our hands on any positive samples for these 2 viruses, so they have not been validated with any samples, unfortunately. That was my presentation.
Okay, thank you very much. I think now we can go on with the Leonics. Arnold Everhardt.
Yes, hi, welcome. Let me share my presentation and go. To switch. I never know where this.
Dig, I think you know where
This one,
it is.
yes.
Ah, okay,
No,
perfect.
Now I see it.
It works.
Good.
Perfect.
Okay, good. So welcome. So the Linux part in this whole project is to make the biosensor photonic chip. What is this biosensor photonic chip? So we have a waveguide here. We have light shining through the waveguide, traveling through the waveguide. The light is mostly confined within a waveguide, but it's a small part of the light actually going out. We call this evanescent tail. And on this part is the biosensor. So on the top of the waveguide, on the top of our chip, we put probes, markers, and that they have a biospecific reaction, biospecific absorption of the biomarkers and the biomolecules that you want to measure. They bind to the surface of the chip. The light senses that we actually have some binding on the surface of the chip, and that's the way we get a signal. So our waveguides, we put them on the chip. We put them with a spiral, mainly to make the spiral to make the surface area as small as possible. Because a spiral is the smallest possible size of the waveguide on the chip, because we want to have a long interaction length of the waveguide with the biomolecules. The longer it is, the more reaction you have, so the higher the signal. Of course, what we need to do for this, and this is a collaboration with our partner Surfix, who's not part of this project, but we collaborate with them extensively on this biosensor chips, is that we have our photonic chips. They make a specific material selective coating, and on this we put the biomarkers. So how does a measurement work? We have here a flow in time with a response. We have a flow of a liquid without a sample. We introduce a sample to the chip. We can see some binding taking place. So this is the binding. We stop flowing the sample, and then we have usually have some dissociation. And this is a typical type of signal you see for the biosensors. And you will see more of these plots and how to exactly do it with the DNA in the next presentation by Curin. So on our part, what we do is we make photonic integrated circuits. So this is a picture of a chip, a photograph of a full chip. We have the light coming in. We have the spirals. So this is one of the biosensors. We are multiplexing it in 6 different biosensors. This way you can run 6 parallel measurements in a single go. And here on the right, I put a picture with actually some red light coupled into it. So you can see that the light is indeed following and indeed lighting up all the waveguides nicely. So here you can see it becomes all red to show the light. Okay, so what we have done, we've built a prototype. So the prototype, of course, it's not only a chip, it's a whole system around it. So the prototype for the complete biosensor, including readout. So here are some optics, the detections, microfluidic box, place where we put the chip in it, and here we actually put a chip. And we have a laptop to control and to control the signals and to follow what happens during the measurement itself. Well, part of this whole setup, the part that we have been mainly concerned about in this specific project, is the chip size. So the chip, and because what happens, you either make a cheap product in the end. The chip price scales directly with the surface area. And we need to have a significantly reduced area compared to what we have at the moment to get to a competitive price for this kind of biosensor measurement chips. Of course, what we do is wafer scale processing. So we process a full wafer. So if we make a smaller area for one chip, we can make more chips for the same cost of the wafer, because the wafer price doesn't change. So we want to make the chip as small as possible to get a reasonable competitive price. So this is what we did in our project to look at the chip dimensions. So currently, the current design, you see it here. You also showed the pictures before. The current design is 50 millimeters square. It's a quite large chip for these biosensors. So what we've done, we focused on reducing the sensing element spirals. So we have a spiral here. We try to reduce them as much as possible. So this is a zoom in of the design of the spiral. And we, in the end, in the SensiChip design, we came to these smaller size spirals. You can see they are actually quite a bit smaller. And since they're also small in 2 dimensions, in the end, we get a quite smaller, much smaller footprint. So a couple of features of this new spiral. First of all, we have a rounder shape. So this is more a square elongated shape. This one is more round, which should also make it easier in the subsequent processing. The waveguides, we put them close together. Here you can see individual waveguides. Here you can see them, they are much more closely spaced together. And we optimized how close can you put the waveguides together without the waveguides basically interacting with each other. So to prevent those kind of signals of coupling from one to the next waveguide. We made the band radius smaller. Of course, if you have light likes to go straight through. So if you go around the corner, around the bend, light typically likes to go straight through. But if you make the bend smooth enough, it will follow the waveguide and go around the circles. So here we reduce the bend radius until we got the point that we actually see the light is losing. We're losing light. So in this sense, we have optimized how quickly can you make the bend. And this is the smallest bend we can make with preserving the light. And what we also did is put the spirals close together, because you can see here they're quite far apart. And here we put them much close together. And we also optimized how close can you put it together, but you can still have proper functionalization afterwards. So in the end, what we managed to get a 3 times smaller spiral surface area, the spiral and the distance between the spirals. So that means a 3 times win of the chip dimensions. And then afterwards you can see, of course, here chip outer side dimension is still the same. It's still 50 millimeters squared. So it is compatible with the current prototype readout system. But it's revealed to optimize all this routing to make it to a chip which is 5 times smaller, so less than 10 square millimeters. And the nice feature of this chip is we actually put a rocket design in here. Also, another part of the project, what we did, is to look at the measurement sensitivity. So the sensitivity of this interferometric measurement, the spirals, it's actually already exceptional and it's really good. So we show biomeasurements below micromolars or nanomolars of DNA, what you will still see in the next presentation. But we need to push it further. One of the ways we think we can do this, we're quite sure we can do this, is to get a 10 times sensitivity improvement by changing the laser, getting a better laser on the whole setup, because we're using light. So because the sensitivity, it scales with the wavelength of the scanning range. So the laser has a specific scanning range of the wavelength. And if we make a laser which can scan at a wider range, we can have a more sensitive chip. So what we did in the project, we designed some chips that makes use of this feature of this bigger wavelength scanning. So what parallel we're already developing, Linux is developing this wider wavelength scanning, which is 10 times bigger than the current lasers that we are using. So we should be able, once the laser is finished, to put the chip on there and directly show they have a 10 times improvement in sensitivity. In the end, what we have to make is a product, and the chip is only a part of the project, the first part of the product, because you need a biofunctionalization, you need a cartridge readout, you need a fluidic integration, and all this has to go into one total product, where only chip is only a small part of this. And within SensiChip, what we have done is to realize a 5 times area reduction of the chip. So we can make chip production with a price which is nearly competitive with the type of biomeasurements we want to do. But we still need to do, and this is not only Linux, this is together with the partners. We need the full integration required in follow-up projects, follow-up activities to achieve the access from 70 for a good competitive price.
Okay, thank you very much.
Okay, I'll stop sharing.
Yes, and I think then we can go on with Floris from Quirin so that we have the whole view on the project.
Okay, thank you very much. I will conclude then this presentation with the contribution of cure and diagnostics. Well, the previous talks have nicely introduced the biosensor and the analytes that are going to be measured with it. Indeed, a small recap, a little bit more comments on the transition from photonic chip to biochip. As shown here, Arnoud has already explained the merits of the interferometers for highly sensitive detection of binding events. Well, for that we need a couple of steps to be done with the photonic chips. For that we Surfix applied for us as a supplier to this project surface selective chemical coating that basically activates all the silicon nitride sensitive areas and passivates the rest of the surfaces, basically thereby focusing the analytes to the sensor and avoiding depletion and increasing the sensitivity. In order to achieve multiplex detection, we use the 6 sensing elements on an interferometer chip for different analytes. So we can apply different bioreceptors, in our case DNA probes, to the different sensor windows using precision spotting equipment that enables the detection of complementary DNA molecules indicative for the different kinds of analytes, pathogens on the different sensors, and basically achieve a interior 6 times multiplexed measurement. The detection of the pathogen DNA itself can be done using the previously shown setup at picomolar sensitivity, as I will show later on. So therefore, the technology as shown here you can see a real picture of a sensor chip with the different droplets representative for the different bioreceptors in order to get a multiplexed biochip. Here, indeed, biochip, and in the setup where we use a little flow cell on top of the sensor that enables control of the fluids, so we can inject a sample with defined volume and do the measurements in that way with the fluidic setup shown here. As already said before, we have different analytes that we are interested in, different pathogens. So in this case, we have asol, vsol, aerosol, virus, which are all bacterial, which is all bacterial DNA that we can detect on the sensor chip, and also a couple of viruses. In this case, we detect RNA on the chip. An example measurement is shown here. There you can see basically a titration experiment in which we progressively increase the analyte concentration in subsequent analyte plugs. So the setup is controlled in such a way that we first condition the chip, then add a low concentration of analyte. In this case, we already see a signal at 10 picomolar concentration of asol. Then we have a flushing step again and increase the concentration to 100 picomolar and 1 nanomolar, etc. And we can see here really well-defined steps in the sensor signal as a response to the different concentrations in the range of 10 picomolar to 1 micromolar, which is really a huge concentration range. And at 1 micromolar, we experience that the sensor shows saturation. But of course, for real applications, we are much more interested in the low range. When we process this data, we see here like a preliminary calibration curve. Yeah, really nice calibration over the range that I just mentioned, from 10 picomolars to 1 micromolar. What we experienced in these experiments is that the different pathogens, the different DNA targets representative for the pathogens, show a different sensitivity on the chip. So this is something that requires further study, but Aeromonas salmonicida, and IPMV showed the best behavior. And you can see indeed that the detection limit for the other pathogens can deviate significantly from this. Another observation that we did, of course, we compared the traditional chips in the beginning of the project, and once the miniaturized designs from Lioness were ready, we also used the new chips. And as a note here, also not only the chip design has changed, but also the chemical coating and the biofunctionalization chemistry in between as well. It has all been further developed and optimized, with a more suitable process for large-scale production in the future. But all these modifications combined, as you can see, result in still quite similar behavior. There can be a factor 10 between the detection limits that we observed when we compare the same DNA target on both chips. But this is really with the low amount of experiments still in the experimental noise, basically. So despite all the changes, we still achieve really good behavior and a really good detection limit on the miniaturized biosensor chips. So as a summary, the primary advantages that are shown in this project of the use of biosensors for aquaculture monitoring is that we can do rapid analysis. I didn't zoom in on the timescales, but an experiment, an analyte injection takes about 5 to 10 minutes. The chips can be made very affordable, especially with the miniaturized design once produced at a large scale. The sensitive detection I showed you before as well. And the multiplexing is possible in this design up to six measurements or 3 duplicate measurements if you're still doing R&D. And of course, this type of micro technology integration enables a much further multiplexing of the amount of sensors in the future. The miniaturized biosensors show similar performance in the preliminary experiments compared to the larger sensors, and with a 5-time reduction of sensor area. We also did direct detection of disease-specific biomarkers in aquaculture, water. Those results I haven't shown here because they are still being evaluated as kind of a final last measurement for the end report of the project. And the picomodel sensitivity has been achieved in the proof of principle experiments. Well, as mentioned, the dependency of the DNA sequence on the sensitivity of the chip is of concern to us, and we have to look into that in order to improve the detection limit for all kinds of DNA and RNA sequences. And the sensitivity towards sample matrices, even though you can measure something, it is still suboptimal. And in the end, of course, you want to use the sensor for a rapid test in raw aquaculture water. And the setup, of course, has to be further developed, as Arnoud also already indicated. Well, for Curin, this project was a very interesting case study, basically, to further work on the biosensors and the setup. But as many people may know, Curin is in the end aiming for cancer diagnostic rapid test. But of course, this is also DNA based in our case, and we see the potential for these biosensor platforms to in the end serve many other applications. So in this case, we worked on aquaculture, but if you're interested in other applications, please contact us, because we can do biosensor based detection in basically every field where a recognition event needs to be measured. So in the end, we can do also proteins or pathogens directly, and the possibilities are endless. So with that, I would like to conclude, and thank you very much for your attention.
Yes, thank you very much. And now I would suggest that we can go over to our questions session. So if you have any questions, put them into the chat or just raise your hand, or I think you can also already activate your microphone and ask your question. Okay, there is a first. What are the next steps for TunerTech and partners? Do the knowledge and the developments within the project provide sufficient confidence to continue to a product? Is a question from Alex. I think to the complete consortium. What are the next steps? What would you say?
Or perhaps to Florian, firstly, is this what TunerTech expected or hoped to achieve? Because it was their question, which surrounded the development. So I was curious, what will they be doing with the results?
I mean, for us, of course, the results are quite interesting, but I think, as Flora stated, there's some more work to do because we need more sensitivity. And I mean, in aquaculture, and that was, of course, the aim of Lyonics, of course, cancer detection in humans, I mean, you can sell a chip for 5,000, 10,000, I don't know, if you can detect cancer within minutes and correctly. But it's hard if you tell your salmon farmer you have to pay at least €5 per fish. Therefore, we shifted to the eDNA, where we can detect, I don't know, 10,000, 100,000 fish at once because they're swimming in the same tank. I think that was quite good that we shifted within the project and within the discussion before starting the project that we aimed on eDNA because we can reduce costs by that. As I said, of course, there's more work to do. I mean, at TunerTech, we are quite lucky that we joined a follow-on project together with Surfix, where I think Kieran is also kind of involved. I've seen Flores in one of the meetings. So I think from our side, work will go on regarding this issue, and I mean that is also fine would be the detection of the pathogens without harming the fish, because it's also always a problem. You can't just go there, kill a fish, slaughter a fish, just to see if there's a pathogen inside. That is, I mean, you have to write an application to do that. So therefore, if we can do it non-invasive, that would be quite good. And if we can do it real time on a chip, or maybe nearly real time on a chip, would be fantastic. I mean, as I said, we already wrote to the state agencies about the multiplex PCRs from NITOR, but right now we don't get an answer yet. But I think all of these state agencies right now, they are measuring corona and not salmon pathogens.
Yeah. Okay. Thank you. Thank you. Interesting.
Okay, I have a question. How do you estimate how long is the way to develop a chip which would be competitive? How much too expensive is the chip until now?
Okay, I can answer this. So at the moment, the selling price for one chip is about €100. So that's, well, it's good for the development phase, not for the final product. If we could reduce the chip size by 5 times, it's 5 times cheaper. So it will be €20 selling price, with a couple of other small optimizations. And if you go to the mass production, we can drive the price down even further. It's a couple of euros. Of course, the chip is not the only part. Chip is only one part of the whole product integration. And in the end, we're aiming for €5 per, well, basically product measurement setup. But you can build it. You have a cartridge readout system, which can cost quite a lot, and then per measurement, you spend €5. That's the end goal of this whole chip production process.
Oh, okay. Okay, sounds good.
Of course, it will take many more years and a lot of investment.
Mm-hmm.
And there are many other projects in the consortium and work going on. It's work in progress. And
yes,
we're definitely getting there.
And
it's not only us.
You mentioned that in the end, this system might be more sensitive than the normal systems who are running right now. So I think in the end, this is possibly another benefit, because then you have a system which reacts earlier than classic systems.
Yes, if we can reach its required sensitivity, yes, that's of course the ideal situation. We are very sensitive and very rapid and cheap and quick. So in the end, it has all the advantages. So it has definitely a high market potential.
So in the end, SensiChip is a very important first step.
Yes.
Okay. Do we have further questions?
If I may ask, for on to Mr. Eberhard from Leonix. So you said you want a different laser, or planning to use a different laser. What's like the main benefit? Why change the laser?
Yeah. Well, I didn't go into the specific technical details. So what we have is currently we use a laser, which is a scanning width of about 3 nanometers. So we're working at a wavelength about 850 nanometers. So it scans from 850 to 853. Well, in that range, you can get a specific sensitivity for the biosensor chip. If we go for a different laser, which scans 30 nanometers, so from 850 to 880, it's a 10 times broader wavelength scan. And we can design a chip that it uses this 10 times broader wavelength scan to make the chip 10 times more sensitive.
Okay, so broader wavelength region is more sensitivity.
Yes, if you design the chip correctly. And that's what we did in the project. We designed chips which use this broadened, well, which is designed to have this really broad wavelength scan.
Okay.
To use this high sensitivity that the laser can provide. But the laser has not been built. We're still building the laser.
That would be, if I may ask another question, like how much of the price or the development is the laser with a larger wavelength area? Is that very expensive, or is it like a cheap comparably cheap diode laser, or?
So the lasers right now is very cheap. It's a few euros for the laser. The laser we're developing is of course more expensive. I'm not sure the price for this kind of lasers, but it's definitely something you can build into a readout system. So you have build a block. You have to buy this whole block with readout system, electronics, and this processing capability, and then you put a new chip every time into this readout system. So you don't have to buy a new laser every measurement, but you can reuse the laser every time. So in that sense, the price of the laser doesn't really matter if it's not exceptionally high, and it's not exceptionally high.
Okay, thank you very much.
Okay, and you also said that the new chip design is 5 times smaller, but you still have no interference between those waveguides, because
correct.
they are much nearer. And okay.
Yeah. So actually, the design we had right now was very safe. We never explored the limits. So this project we actually explored the limits. How close can we put it together that there's no interference between the waveguides? So we actually managed to put them much closer together.
Okay. Okay. It's great. So I think in the end you could be you could even create a even smaller chip, but it's not useful.
You could create even smaller chips, yes.
Okay.
Of course, we set the benchmark of this is going to be the rough chip area. You can shave a couple of things off still. It's not the final chip design, and we're constantly improving it.
Yes, but 5 times smaller is a great result in the end.
We did a big job in 5 times smaller, yes.
Yeah. Okay. So, and with the okay. Okay, I will, yes, next week we will have another webinar for Automath 3D. I will come back to this later on, but there are, Dirk, we have another project about photonic integrated circuits, but it's another material basis. What exactly is the difference between those both? Indium phosphide and silicium?
Gallium arsenide.
Gallium arsenide, okay. But the system you are using is more common. Is that right? And therefore,
I'm not saying one is more common than the other one. So there are two different systems. We're using silicon nitride. So this is, well, it's an insulator. So it's a full passive chip. And the advantage of the silicon nitride, it works in the visible region and is extremely low loss.
Okay, great.
So we lose barely no light. Well, silicon, the indium phosphide has other applications. It's an active material. You can do it has inherent active components, but it works only in the infrared and has much higher losses. So there are two systems, two different for waveguides. They both have advantages, disadvantages, and basically a kind of different application areas and also the same application areas.
Okay, okay, I know. Okay.
If I may add a short question, so is the silicon nitride actually coming into contact with the, or is this sensing? There's a buffer where the sensing is.
It comes in direct contact. Well, okay, almost direct contact because we have a coating on top of the silicon nitride. So this coating provides the biofunctionalization, but the coating comes in direct contact with the silicon nitride.
Okay, so would it be like also a benefit that silicon nitride is super chemically inert, maybe for this? I'm not sure how corrosive or how stable gallium arsenide, so for example, would be, but you never want an arsenide compound in an aqueous system.
You do not want to gallium arsenide. Well, they're solid materials. They won't lose atoms of silicon gallium arsenide, but definitely it's a safe material. You can put it in a fish. Well, you could even put it in a fish, yes, without harming the fish. So it's definitely safe.
And the biomarkers, this is the biofunctionalization. It's just the coating. So I think it depends on the coating which DNA you are detecting. That's right?
Yeah, there's the coating developed by Surfix. They have a specific coating, and on top of this coating, you basically apply your biomarkers. You could choose a specific biomarkers, as Floris showed in his presentation. You see the drops on each of the spirals. Each of the drops can contain a different biomarker. So each can detect a different DNA sequence. So you can have 6 parallel, completely different DNA measurement units in the same chip. So
or already done systems. Okay.
Oh yeah,
that's great. So, but in the end, this makes your solution very versatile. So you started with fish farming, but you can use it for water quality, groundwater, for everything in the end. That's right.
Exactly
because it's just the
said.
coating. The system stays the same, and you just change the coating, and then you have another application field.
Exactly what Floris said in his presentation. There's so many application fields for this whole technology.
Great. Okay. Thank you. Are there further questions? I think not now. But as I think already mentioned, we made a screen recording, yes, and we will put it on the website so that all the participants and also additional guests can take a look at it and then get into contact to you. Thank you very much for your presentations and your time for answering our questions. And then I would I would like to tell you, next week we have another Rocket Reloaded webinar. This is on Automath 3D. The consortium developed automatic measuring and analyzing system for shape and positional tolerances. So today was more about biotechnology, and next week we have engineering, classic engineering, I would say. So maybe if it's also interesting for you, you may join us. Thank you all for your participation, and have a nice evening. And I think I will send the link with the video as soon as we have the release, the permission to release it. Thank you very much.
Thank you. Bye-bye.
Bye-bye.
Bye.